Is this working? No. Let's wait. Yes? Okay. Thanks for the introduction, Doug. And also thanks the organizer for giving me the opportunity to present my work here. So today I'm going to talk about amyloids. So I'm sure that many of you know what an amyloid is. But in any case, I want to say that an amyloid fibril is a very hard protein aggregate that has this beautiful shape and are characterized by the cross-beta structure that consists on monomers with beta strands that are one over the other. And they are also widely known because they are pathological hallmarks of more than 40 human diseases. However, something puzzling about this structure is that it can also be functional. And actually in nature, we have many examples of functional amyloids that play many different roles. And also in the last years, artificial amyloids have been developed to fulfill different functions in the industry. Despite the advances in this field, there are still a lot of open questions. And one of them is the role of functional amyloids in necroptosis. So what is necroptosis? Necroptosis is a lytic and inflammatory form of cell death that is triggered upon stimulation with a molecule, for example, as tumor necrosis factor alpha. Then in a first step, we have RIPK 1 activation. And later, this kinase interacts with other kinase, that is RIPK 3, through the rim domains that you can see here. These rim domains oligomerize, forming this amyloid fibril that serves as a scaffold for the necrosome complex that recruits and activates MLKL, that is the final effector of the pathway leading to cell death. So, what are the differences between functional amyloids and pathological amyloids? How are these functional amyloids formed? What are the impacts of mutation in the rim domain? And what's the interplay between amyloid nucleation, amyloid aggregation, and necroptosis, and the functional outcome of these enzymes are some of the questions that we have in the lab. So to answer this, we decided to demutagenize both rim domains, the rim domain of RIPK1 and the rim domain of RIPK3. These rim domains can form homo-oligomers that leads to an amyloid fibril or hetero-oligomers. So we focus now on the homo-oligomers. So for doing this, we have this classical demutational scanning pipeline, starting with a library, in this case, library of variants of RIPK1 or RIPK3. Then we select by amyloid formation, and we end up with deep sequencing to calculate our fitness score. So what I want to stress here is that we are selecting on the basis of nucleation of amyloid aggregates. And as we know that our fitness score is correlating with the kinetics of amyloid aggregation, with this I mean how fast these amyloids are formed, we decided to call it as nucleation score. So we started mutagenizing the rim domain and flanking regions of RIPK1. As you can see here in these heat maps, there's a lot of information condensed. We have some mutations that decrease nucleations, are the one in red. Then we have some others that increase the amyloid nucleation, are the ones in blue, and some others that are wild-type-like, that are the ones that are in gray. Then we also mapped the median nucleation score per position in the monomeric cross-section of the amyloid fibril of RIPK1. As you can see, this is an S-shaped fold where we can distinguish 3 beta strands and 2 interfaces. And with our deep mutational scanning, we identified this aliphatic tetrad where most of the mutation decrease nucleation and these residues are forming the first interface. Then this triad of residues that where also most of the mutation decrease nucleation and are forming the second interface. And then this glycine here just before the second beta strand where most of the mutation increase nucleation. Then we also mutagenize RIPK3, the rim domain, and the flanking regions. And again, we have some mutation that decrease nucleation, some other that increase. And we also identify here this aliphatic tetrad, where most of the mutation decrease nucleation, and these residues are forming the first interface, and also the glycine residue, where mutations increase nucleation. So apparently, this first interface is a shared feature between these two sequences, but we can clearly see here that in the second interface, the effects of mutations are quite different. while in RIPK1, most of the mutation decrease nucleation. In RIPK3, we have no effects or even increasing nucleation. So we also wanted to compare this in a more quantitative way. So here in this scatter plot, we can see that those residues that are forming the rim domain has a moderate correlation, while those that are outside the rim domain, there's no correlation for them. This is also a correlation of the effect of mutation, but taking into account the different structures in this monomeric form. And as you can see, we detected this positive correlation starting from beta strand 1 until loop 2, but this positive correlation is lost in beta strand 3 and in the disorder region. So with this, we identify what is shared between RIPK1 and RIPK3 and what's different, right? So, but what's the effects of mutations in sequences from other species? So we decided to also demutationize the mouse orthologs, and we found that the aliphatic tetrad, that are these residues, the effect of mutation on the aliphatic tetrad is conserved, as well as the glycine position, and also the difference in the interfaces is conserved as well. So we also made use of evolutionary scores, in this case, GEM score. If we do this correlation, like taking into account the whole sequence, the effect of mutation in the whole sequence, we cannot see any correlation, or the correlation is very, very poor. But if we do this per position, we measure the Pearson correlation between our nucleation score and GEM score per position, we start seeing that at some positions we have significant correlation. And here I'm highlighting the aliphatic tetrad that I already told you. And in particular, you can see that in RIPK3, we have positive correlation that is significant. So this is telling us that this aliphatic tetrad that is key for amyloid nucleation is evolutionary conserved. However, this evolutionary score, can detect some important residues for amyloid nucleation, but cannot capture all of them, as for example, the glycine that is just before the second beta strand is not well captured by this type of evolutionary scores. So, but what's the relationship between amyloid nucleation and necroptosis, the functional outcome of these enzymes? So for this, we started searching in the literature for some evidence, some measurement of the effect of mutations on the rim domain in the kinase activity or in the necroptosis pathway. And we mainly find this effect of mutation on cell death, as you can see here. So you can clearly see that we have a positive correlation between our nucleation score and cell death. But also here, we can see that most of these mutations decrease nucleation are wild-type-like. Until we found this piece of data that are RIPK3 mouse mutants, where we also have these 2 points here that are mutations that increase nucleation. In this case, the authors measure cell survival instead of cell death, so this is the other way around. The wild type is here instead of being here at the top, right? So what is interesting from here is that you can see that there's like a quadratic correlation. So in either way that you disrupt amyloid nucleation, by this I mean if you decrease or if you increase amyloid nucleation, you are also disrupting necroptosis. So thereafter, we wanted to know if there's some evidence of this in the human population, and we went to NOMAD and retrieved from there all the variants that are reported, and what we found is that those variants that are disrupting amyloid nucleation are extremely rare in the human population, while those that are more frequent are wild-type-like. So this data seems to suggest that a sweet spot of amyloid nucleation is needed for necroptosis. So of course, this data is very interesting, but we want to do this in a more systematic way, and we also want to correlate amyloid nucleation with necroptosis, but also with fiber stability. So we need to scale this up a bit. So we can measure the effects of mutations in nucleation with our nucleation assay, as I already showed you. We can also measure the effect of mutations in fiber stability using a software such as Foldex, but how can we measure necroptosis at the scale? So for this, we use a reconstituted necroptosis pathway where we can measure mutations in the rim domain of RIPK3. the effect of mutations on the ring domain of RIPK3. So here we are measuring the effect of necroptosis of the same library that we scan here. So by doing this, we can ask what's the relationship between these three features. So here I'm showing in the y-axis how stable these mutants are, in the x-axis how fast they are, so the nucleation score, our nucleation score, and the colors are the effects of these mutations on necroptosis. So in green we have decreased necroptosis, and in black we have wild-type-like necroptosis. So we have this V-shaped plot where most of the wild-type-like variants are significantly enriched in this region, while outside this square, this tile that is around 0, we have many of the green dots. This can also be seen here. What I'm plotting here is the distance to the origin of each of these dots. And as you can see, the green distribution is right skewed and is shifted to the right also in this plot. So this data is also supporting our hypothesis of the sweet spot of amyloid formation that is needed for RIP kinase 3 activity and necroptosis. So in conclusions, the rim domain of the RIP kinases contain an aliphatic tetrad that nucleates the formation of the first interface in the amyloid structure. Substitutions of the glycine located before the second beta strand lead to an increase in amyloid nucleation. The impact of mutations on RIP kinase 1 and RIPK3 nucleation differs in one of the two core interfaces of these amyloids. RIP kinase 3 activity and so necroptosis needs a sweet spot of amyloid formation. This is reflected in the allele frequencies of RIPK3 variants. Variants affecting nucleation are extremely rare. And finally, I want to highlight that DMS is a powerful tool to establish the relationship between amyloid formation and amyloid function, and this is useful because it helps us to answer the question that I have at the very beginning of this talk. And it's also very important because it helps us to guide the development of new therapeutic strategies and also for engineering active amyloids. So finally, I would like to thank all my team, especially Benny Bolognesi, that is here, is one of the organizers. So also thank you to you, and I'm open for discussion. Thanks.